Welcome to everyone who joined the webinar today. As David said, we are going to talk about MobiDB, which is a database that provides predictions and annotations for intrinsically disordered proteins. So this is the homepage of MobiDB, and we are going to describe it in a while. But first, I would like to show you that here we have a version of the resource, which is version 4.1, which is the release, the number of entries, and the most recent publication that describes MobiDB. First, before talking about the homepage itself, I would like to talk about the About page of MobiDB, which is the documentation page that you have to check in order to understand all the features, all the evidence levels annotated in MobiDB, and to have a better comprehension of the database itself. So as we mentioned, MobiDB provides annotation and information about intrinsically disordered regions from various resources and the prediction tools. You can cite the database using the reference here, which is our latest NAR update in 2021. And you are going to see today different type of features in MobiDB. For example, you are going to see one of these type of annotation, which is the evidence. It is the evidence level. That could be something that is manually curated. So from manually curated resource, it could be a prediction derived or an homology annotation. The feature, which is the type of annotation, let's say disorder, missing residues, for example, binding mode, or linear interacting peptide. And also the source. The source is the source of the annotation. It can be a third-party database. It can be a software, for example. So we have some examples here, MobiDB Lite, Dept for curated annotations. And you also have different levels for the evidence in MobiDB. You can see here that they are characterized, they are characterized, each one of them by a specific color and letter. We have curated here, so manually curated experimental annotations from a partner database, which could be Dept, for example, a derived annotation from primary data, PDB. For homology, so annotations that are derived from aligning curated regions against ensemble. And the predictions, so the predictions are annotations that are provided running softwares on against the UniProt proteins. So these are some of the main things that you have to keep into account when we are going to explore MobiDB. MobiDB has also different type of consensus. You can see here, strict, majority, merge, or priority. And all these different levels are described in the About page. Today, we are going to have an introduction to the resource, and more detailed information are going to be covered in a second webinar that is going to be an intermediate level webinar about the resource. By exploring more the About page, you will also be able to find the main features here. So information about the intrinsic disorder that is annotated in MobiDB. So the sources of this information that could be from manually curated databases, missing residues, so X-ray derived from X-ray structures, mobile residues, so intrinsically disordered regions from NMR structures, and also the MobiDB Lite predictions. And also you can find more information about the LIPs, the linear interactive peptides, again, the binding modes, and a features table of all the features that you can find in MobiDB for each one of the categories, may it be the binding, the disorder, or interactions, for example, or other features too. So here you will see for each category the features and the source and the evidence that you can find for each feature. Also the consensus level that we have seen a few seconds ago. So if we look now at the MobiDB homepage based on the information that you already have seen from the About page. As I mentioned before, we have some general information like the version of the database and the release, the number of entries. You can perform a search here, and we will do it in a while. So we will also explore an entry in MobiDB. This is the latest publication. So what about the information you can find in the homepage of MobiDB? You can find manually curated annotations. So these are the databases from which MobiDB retrieves its manually curated annotations. Each color of a database means a different feature. Red is for a disorder, has a feature. The purple is a linear interactive peptide, and green is binding of a disorder region to another disorder region. And also we have light blue that represents the phase separation. So if we look, for example, here in this plot, what can you see for each one of these databases? You can see the number of annotated proteins in MobiDB and the percentage of annotated residues over the total number of residues. But you can also, for example, open the source itself, and you can download the JSON file with all the proteins that are annotated from that specific resource. This you can do it for every one of our manually curated annotation sources that are shown here. So for example, you can do it for DisProt, Ideal, Dept, and FacePro. Then we have the derived annotations that we have seen a while ago. And again, here, the color of the icon represents the type of the feature. It could be mobile, it could be binding of disorder to an ordered region, or missing residues. As we have seen before, mobile residues are from NMR structures and missing from X-ray structures. Again, you see the number of annotated proteins and the percentage of annotated residues, and you can download a JSON file of all the proteins from that from those derived annotations by clicking on this button here. Also, you could click here and search for mobile residues proteins in MobiDB, and it will open a dedicated page with all the entries that are annotated with that feature. Finally, in the second half of the homepage, you will find the organisms. We have eukaryotes, bacteria, and viruses. So for what concerns the eukaryotes, you will find some of the most common model organisms, such as zebrafish or mouse, or for example, the fruit fly and Arabidopsis thaliana. You can also find several other organisms. These are not all the organisms that are described in the database. There are many others too that you can explore from the browse page. What you see for each one of these organisms here. Let's look at the zebrafish, for example. So you can see the page with the full annotation statistics for the proteome of zebrafish, and you can search for all the proteins of zebrafish in MobiDB here by using these two buttons. What are you seeing here is the curated, derived, predicted annotation for this organism, again with the percentage of annotated residues and the number of annotated residues, annotated disorder proteins in a specific with a specific feature. So for example, we could click here now, and you will see the full annotation statistics for zebrafish. This is the page. You can see here that you can perform a search. Okay, in this case, it was performed against the proteome of zebrafish. You can see here the identifier. And this is the table with all the features. You can sort it by evidence. For example, let's say that you are more interested in prediction or you're interested in curated. You can sort it by feature, which could be several, could be transmembrane, signal peptide, phase separation, for example. You can also use the source An example could be UniProt or FacePro. And here again, you have all the information for each one of these evidence and these features in the database. So the number of proteins, the number of residues, the total number of residues for the annotated proteins in MobiDB, and also the fraction residues and proteins in the proteome. And for each organism, when you look at the dedicated statistic page in MobiDB, you will see the number of proteins that are available in the database for that specific organism, the number of residues, the proteome identifier, and the NCBI taxon identifier. You can, of course, download all these statistics using the button here. So going back to the home page, the organism, so you can do the same for each one of the organisms that are shown here. As I said before, you also have bacteria, and you have some very common bacteria here. We have viruses now since last year, as we have SARS-CoV-2 information too, curated also, but also derived and predicted. And you can look for other organisms by using the statistics page of MobiDB. So as I mentioned before, you can look for a specific protein in MobiDB. What I recommend is that if you're interested in a protein from a specific organism, you could also use the UniProt accession number instead of looking for the name of the protein, so that you find the correct entry for that specific organism. So now we are looking for the ATG3 protein from Saccharomyces cerevisiae, and it's here. And this is autophagy-related protein 3. As you can see, this is the UniProt identifier of the protein. The gene, so here in those info cards, you will see the general information about the protein, the features content, and if available, the interactors. Then we will have the feature viewer and the cross references to other sources for this protein. So here you see the name of the gene, the organism. As I mentioned before, we were looking for this organism, the taxon identifier, reference proteome, and the dataset from which it's derived. In this case, it's Swissprot, and also a localization of the protein. Here you can see also the interactors. ATG3 is interacting with ATG7, another autophagy-related protein. And you can see here the features content. So you have disorder, linear interactive peptides from different sources and domains. Before talking about the feature viewer, I would like to show you the cross-references here. So at the end of the page, you will see the cross-reference of UniRef, the reference proteome, ELM and Disprot, ELM for short linear motifs, and Disprot for manually curated information of intrinsically disordered proteins and regions from literature. And then we have a cross-reference to PFAM for the domains, UniPark and AlphaFold. So indeed, this is the feature viewer. You can use two different modes. One is the simple and one is the advanced that can be customized based on what you're looking for. So in the simple version of the feature viewer, you're going to see that we have the whole sequence of the protein that is slightly more than 300 residues, and the disorder consensus. The disorder consensus based on MobiDB Lite and AlphaFold here predicted, and the LIP consensus, so linear interactive peptides. You see here that the regions in red represent the disordered regions. So if you scroll over a specific segment on the feature viewer, you will see which is the corresponding region that is described. So here it's region 120 of the protein. Again, here we have another region that is disordered from residue 84 to 162. Again, another region here from residue 247 to 280. And you can see that these regions are also corresponding to the regions with a low PLDDT lower than 50 or 70 in AlphaFold. If you want, you can also show or hide the sequence viewer when looking at the feature viewer. So you can see here highlighted on the sequence each one of the regions you're interested into, okay? If we want to move to the advanced mode, what we can see here is several more information, but still you can show or hide the sequence viewer, which could be useful to highlight features as PTMs. So here we have an acetyl lysine as a post-translational modification. Again, here we have another acetyl lysine as post-translational modification. We have the disorder consensus that we have just seen and the curated disorder consensus. This information is derived from Dept. So if you look for this entry in Dept, you will see that these are the regions that have been manually curated based on literature experiments published in scientific papers. But you can also see the missing residues content and the disorder consensus based on prediction methods. So these are the different prediction methods here, and this is the disorder consensus. Again, here we have low complexity consensus. And here you have the method seg. Finally, you have the information about AlphaFold that we just have seen, linear interactive peptides, curated or derived, and anchor, which is predicted, binding mode consensus, domain consensus, so PIFA. Again, here for binding, we have MOBI, which is derived, and the interaction consensus, that is another derived feature. So you can look for all these regions into the sequence viewer tool. And you can find more information in this part of the page that is shown here. So you will see if there is a selected interactor, if there is a selected structure, if you have a PDB structure, which is the selected region. In this case, we have no interactors in the region that we have selected. So this is a page of MobiDB, an entry page in MobiDB. And you can also customize the feature viewer because you may want to hide some of this information, such as, for example, the lib consensus, the binding mode, AlphaFold, or the domain consensus. You just have to unflag the corresponding checkbox. And you can finally download the entry, the information of the entry in 3 different formats. You can choose from fast.json or a TSV file. Then we have seen before that we have a statistic page that we already explored. We have an help page with the API documentation if you want to programmatically access the database. So here you can find all the information to search entries, to download the entries, the statistics of the organism, and the query parameters for the search and download. And you also have information about the output download, so the 3 different file formats that are available for download in MobiDB, which are the JSON file, TSV, and FASTA file, with examples of how this file look like once you download them, and the examples here. Finally, we have a release notes page, and this is where you can find the information about the different releases of the resource, or about the different versions of the sources of annotation that are available in MobiDB. So you can see for the most recent version of MobiDB that was released in December 2021, we had these versions that were used for each one of these resources. So you have the version for Uniprot, PDB, Ideal, L, and so on. Here in the release, you will see all the releases from the newest one to the oldest one, and you will see the which is the version of the release, when it was released, and what was done. So it could be a technical release, or there could be some updates on the graphical interface or on the feature viewer. So all this information can be found here in the release notes. And that's it for this introductory webinar on MobiDB, and we will have soon an intermediate level webinar on the resource. Thank you.